I'm Wasim. I'm a solutions developer at Align to Innovate.
I'm Erica. I founded Align, and Wasim is going to walk us through how to onboard primers from IDT into Emerald Cloud Lab, which is a fast introduction to what it's like to work in the cloud.
That sounds great. Thank you for the introduction, Erica. So, for Emerald Cloud Lab, you basically work with this software that they provide called the Command Center. It's a tool where you access every single aspect of their cloud lab. And as a first step, in order to perform any experiment for your DNA pipeline, you would first need to order the DNA samples and reagents. Then you can, once you have those in the physical lab, then you can use them to perform experiments. So today we are going to take a look at how to actually do that using the Command Center software that they provide us with. So when you actually open the software, you're greeted with this screen where you will have a bunch of notebooks and this dialog up top with different buttons that you can scroll through, which will give access to different types of commands and different types of functionality that they have. In ECL, we're going to talk about today is the first step of performing your DNA manipulation, where you're ordering primers, DNA, and reagents. In order to do that, you'll have a notebook open, or you can get a copy of this script from our GitHub link. And this is a slight variation on what you have on GitHub, but this will explain you better as to what's happening on the backside or actually happening as the script runs on the command center. So in order to actually have any sample on ECL, there are three different steps. First, you have to create the identity model, which is basically an idea of a molecule that you create on their platform. And then using that molecule, you can think of it as like a component, and use that component to derive something called a fulfillment model. A fulfillment model is you can think of it as like an idea of a sample, be it like a DNA, be it like a protein, be it like a reagent, be it like a chemical. It can be anything. And that's the idea of that sample or chemical can be defined using the first identity model that you created as like a building block. And you can use that identity models, maybe one or multiple of them, in different composition to create the idea of a sample. And then once you have the idea of that sample, you can create or order your sample from the respective maybe vendor, or you can even like ship it out yourself to Emerald Cloud Lab.
So can
So
I jump in? Just
absolutely.
so like one of the things that I think is surprising to folks who are accustomed to working in person is just how much this particular Cloud Lab system knows about every single sample in your inventory. So Emerald tracks the chemical composition of every liquid you have, and every single different little drop of liquid has a sample ID. And because of this system is able to kind of reason about chemistry and like what happens when you mix things, which is cool. But the flip side is that when you do something as simple as ordering a primer, there's an enormous amount of detail you have to give to the system before it can work with it, because it has to know, like, literally down to the atoms, like what is in that primer chemically.
Absolutely.
And so that's what happens here. So you have to go all the way from the molecules that are going to be in your sample to the idea of the mixture of molecules that's in your sample, like solvated DNA, to how do you actually produce that sample, to this here is a tube that has this sample in it. And you get all the way from the idea of the molecules to a physical sample that contains that DNA. And so that's what this script is for.
That is true. Thank you for that input. Before we jump into like how to order the DNA itself, I'm going to show you an example of like a hypothetical chemical that we are ordering on ECL, just to drill down the idea of creating the identity model followed by the fulfillment model and then ordering the sample itself. For my example here, I'm taking a model of methanol itself. Fortunately, ECL already has an identity model or the idea of a methanol molecule itself. If in case the chemical that you're trying to create doesn't have an identity model, you can easily go into the commands and use the function called upload molecule to create the identity model of a particular chemical or a molecule. Fortunately, if you already have this
That
identity
and
model,
that model, it knows things like the chemical structure, right? So if you were
that
to
is
like
true. So
inspect,
if we were to see what exactly what all the information is contained within this identity model, I am doing a quick overview of the information that might be present in here, and that is to inspect that particular model. And as you can see, it will have the chemical structure of the model, what different synonyms are used, who created it, um, what's the PubChem ID, the CAS number, uh, and all of these different properties of that particular molecule, including
right? This
molecular
is
weight.
where like the chemical knowledge
This
lives
is
in a
all the
system.
chemical knowledge that is contained within the identity model itself. And yeah, while creating it, you can also provide like a safety sheet that the vendor provides you with, and all of these information of how to handle it properly in lab, if it's like a hazard or not, and all of that cool information is contained in here. And once you have the identity model itself, you can then use that to create this something called the fulfillment model, which is the idea of a sample itself, where it can be composed of one or two components or maybe more components. For our case here, it's composed of 99.9% of the methanol molecule, while 0.1% we do not really know. It could be impurities, so we are providing it as like null and null. So at least the system knows that there is something that it doesn't know about of 0.1% present in this idea of a sample. Once you have that compiled and run, you'll get the model sample, which is the idea of the model. We can actually inspect this one too to see what information we have provided or given to this whole object. As you can see, it can take information from the model molecule, the identity model itself, and populate a number of fields from within. So it has calculated a bunch of parameters, physical properties. It also got the safety sheet from the molecule itself, the biosafety level, the handling, and everything else. And also, if incompatible materials are informed, it'll also populate this based on if there are one or more identity models involved within a sample model. So now you can think that we have the idea of a sample now, but how do we actually order and get the physical sample in lab? In order to do that, you would have to actually place the order, but the system doesn't know where actually to place the order. In order to do that, you have to actually create a product linking the website or the link from where it can be ordered, and like the description, the price, and everything in order for ECL to track how much money you are spending on their platform to order these different chemicals. And once you have the upload product, you will get the object product, which is an object that the order samples takes information from in order to place the order for the sample itself. So here, finally, we are now ready to place the order for the samples. You give the product itself that you associated with the model sample. So within the product, you're also associating this with the model sample that you created earlier. You can give that product and how many of that product you want to order. In return, you will get the transaction ID, and this transaction ID will track if your items are received. And finally, this ID, object sample ID, will give you the actual physical sample that is present in the lab.
So basically, if you want to order something from the internet, you have to define it as a product.
Mm-hmm.
You have to work backward and you have to create a fulfillment model that describes here are all the things, chemically speaking, that are within this thing that I could order from the internet. And then for each of those chemical things, it actually has to know the chemistry, like it literally has to
Right.
know the chemical structure, or you can put null.
Yes.
And then you can place an order, you can track whether that order has arrived, and when it does, you'll get sample IDs that correspond to this here tube of HPLC grade methanol.
That is true.
Can
Yes.
you see if those ones have pictures
Um, those are actually of
not?
different samples. Maybe I'm going to show you the samples for the DNA
Okay,
itself,
very
so
good.
it'll be much easier
Yeah,
that way.
let's go to DNA, and the ordering DNA is a little more complicated because you have to define the molecule
Right.
yourself. So one extra step there.
That is true. So, like Erica mentioned, we have to define the molecule itself, and for DNA, it's slightly more complex because you have to define the sequence itself. So once you know what your sequence is, you gotta give it to the function called upload oligomer and provide it what type of sequence it's gonna be, and it's gonna create you an identity model for that single-stranded DNA.
And that if you inspected it, it would show you the strand, right?
Yes, it will show you the strand.
This is where again the literally the chemistry of it is tracked.
There you see. It will
Yeah.
show you the strand that you just inputted.
Hmm.
And it'll have all the chemistry. It automatically calculated the molecular weight, extinction coefficients, and everything
Hmm.
that you need for this particular oligomer. Once you have the identity model of the oligomer itself, you can then create the fulfillment model for that primer using the oligomer. I'm giving 100% or 100 mass percent of this ID of a sample is containing the oligomer molecule itself. So all of it we know is the oligomer here. And then I'm providing a bunch of properties, physical properties, how it should be handled, where it should be stored, how long it's going to be stable on the shelf, how long after it's going to be expiring and stuff like that. And if
And
it,
this is where like if you were gonna order pre-resuspended oligos
right,
from IDT instead, you would not specify it as a solid powder. You would specify it as liquid, and you would, in addition to saying it has this here DNA chemical in it, you would also say it's solvated in water.
yes. So you'll have to kind of specify the concentration of your oligomer followed by the diluent itself or the amount of diluent there is. If it's water, you got to specify like 95% of the sample is water, the rest is oligomer, and so on. That's a great point. And once you have the fulfillment model, you can, I'm going to just scroll down this bit a little for now because I want to show you how you're going to place the order. For placing the order of the DNA from IDT, it's a slightly different process than just ordering samples because on the IDT website, you have to populate the information yourself because ECL cannot populate the sequence information and everything and then place
Yeah,
the
it's
order
not
for
like
you.
there's no standard part number.
There is no standard part number for each and every custom created oligo. So you have to enter that information yourself, provide that information to ECL, and they have already created like an object for the company service IDT, so they know who the supplier themselves are, and then once the order has been placed, the reference number can be used to track the order using dropship samples. And here is the fulfillment model that we just created moments ago. That was for a single-stranded DNA. For the double-stranded DNA, it's a slightly more complex process, but bear with me. It's going to be clear in a second. So for the double-stranded DNA, I'm here defining the sequence itself first, and then using that sequence, I'm defining the structure of the DNA itself, where I'm taking the DNA strand and the reverse complement strand, and then kind of combining them together in like a double-stranded way to create this idea of a double-stranded DNA. And using that double-stranded DNA, I can create that identity model of that double-stranded DNA by passing in the molecule structure we just created. So it's like a one step lower than what we did for the oligomer itself. Once you have the identity model, you can use that to create the fulfillment model, again, using 100% of that identity model because we know that it is going to be a lyophilized solid form that we're going to receive from IDT. Again, in the end, we're going to again receive the fulfillment model, which we can provide to the dropship samples to track how these samples are ordered and received at ECL's facility. Once this command is run, you're going to receive a transaction dropshipping, which is basically an object to track where exactly is your order. And once it has been received, you can get the received items using this field called received items within the transaction shipping. And here are your actual samples that have been received, and we can take a look at how they look like. Here is
There we go.
your final DNA tube that was received from IDT.
And, IDT will attempt to synthesize at, some synthesis scale, but then the amount you actually get gets written on the tube and in the paperwork and stuff like that.
Yeah.
And when someone receives it, I think they end up
They will populate all of that information that they receive from IDT. So you don't have to worry about specifying the amount beforehand. You can here, as you can see, mass, I have given it as product documentation. So whatever documentation they get along with the order, they're going to populate that the mass information from that product documentation.
Cool. And
So,
does this script have an example of resuspending, or is that in a different script?
I have an example of resuspending here too, so
There it is.
that's perfect.
Great.
That's a great segue into that. So once you have received those primers, I have assigned the primers to a variable called primers. So primers would be, let's see, yeah, these are the two samples that are primers, and then I pass it on to experiment resuspend, and I'm giving it a target concentration of 100 micromolar. So I want a stock concentration of 100 micromolar using the TE buffer, and I want them to roll it to resuspend, and after resuspend, I want them to wait for 5 minutes and then store these samples in a freezer. And what you get out of it is an experiment protocol or an object protocol sample preparation. And then after the experiment is completed, you have the same samples, and then you can actually check the volume. Here, the volume is given in liters. You can actually convert it to microliters and see how much volume is actually present in them at this current moment.
Cool. Thanks, Wasim.
Thank you so much. Thanks everybody for listening, and hope you have a great time creating and ordering samples on ECL's platform.
Have fun with your primers. Cool.